Welcome to the first video of AP with Miss C. Miss Cote here, and I will be your guide through this wonderful and frustrating class called AP Biology. In this episode, we'll be reviewing carbohydrates. After reviewing the basics of chemistry from our summer assignment, we're ready to jump into our first unit of AP Biology, which is biochemistry. So we're going to see what happens when we take atoms of different elements and build some large structures called macromolecules. Just a disclaimer here, I assume that at some point you've all taken a basic biology course before coming to AP Bio, and I may occasionally refer back to what you should have learned. So if you haven't taken bio before, or if you did and it's been a while and you don't remember what I'm talking about, then you may need to work a little harder to build up or refresh your background. But don't worry, it's totally doable. And to help entertain you, some of my former students created a Bitmoji version of myself to insert in various places. I think it's pretty cool, but just let me know if I start getting too obnoxious with it. Anyhow, back in biology class, you should have learned about the 4 biological molecules: carbohydrates, lipids, proteins, and nucleic acids, also known as the macromolecules. You learned about the elements that make them up, their monomers, their polymers, their structures, their functions, and some relevant examples. So this should be a nice review for you, with a little bit more detail built in, because this is AP after all. One of the recurring themes of this class is that structure is related to function. You'll see as we explore these biological molecules, how they're built will give them certain properties, and that will determine their different functions. Before we dive into the macromolecules, let's review some vocabulary. Hopefully you remember that these macromolecules are considered to be polymers, made up of repeating subunits called monomers. So if you think about the 4 biological molecules, only 3 of them by definition are true polymers. And if you can't figure out why lipids are not on this list, well, we'll talk about that when we get to the lipids video. You should also be familiar with the processes that build or break down macromolecules. The name of the process that builds a polymer is a dehydration reaction. That's what our book calls it. But know that it can also be referred to as dehydration synthesis or a condensation reaction. Whatever you call it, this process adds a monomer to either another monomer or a growing chain of monomers, and this makes a polymer. And in order to do that, a molecule of water is released during the reaction. One of the monomers provides a hydroxyl group, while the other provides a hydrogen, and you put them together, and bam, you get a molecule of water. To break a polymer down into monomers is the opposite reaction, called hydrolysis. Hydrolysis literally means splitting water. So you take a water molecule and you split it up, with hydrogen attaching to one monomer and the hydroxyl group attaching to the other monomer. And this breaks the bond between the monomers. Now that we have the basics down, let's start with the carbohydrates. Carbs are your sugars and your polymers of sugars. You've probably learned that most carbohydrates end in OSE, but there are always exceptions to the rule. The simplest sugar or monomer of a carbohydrate is a monosaccharide, which literally means one sugar. Monosaccharides serve as a source of fuel for cells and are also used as raw material for building larger molecules. 2 monomers can be joined together to form a disaccharide, literally 2 sugars. And many monomers can be joined to form a polysaccharide, again, many sugars. But before we get into that, let's take a closer look at monosaccharides. Carbohydrates contain the elements carbon, hydrogen, and oxygen, and monosaccharides have fixed molecular formulas that are a multiple of CH2O. Back in biology, I know you studied glucose quite a bit, especially with photosynthesis and cellular respiration, and hopefully you remember the molecular formula being C6H12O6. See, it fits into that general formula. But please know that there are other monosaccharides out there, and they can be classified in 2 ways. The first way is the location of the carbonyl group. Remember that functional group, it can be a ketone or an aldehyde. And second, how many carbons it contains. So let's look at some examples. Here are examples of carbs classified by the number of carbons. Here we have some 3, 5, and 6 carbon sugars, and also classified by whether it's in the aldehyde or ketone form. Remember from studying functional groups in Chapter 4 that aldehydes are carbonyl groups found at the ends of structures, while ketones are carbonyl groups found in the middle of structures. And because these are sugars, you may also see the terms aldose and ketose, note the OSE at the end of the name, to identify that. Another means of classifying carbs would be using spatial arrangement of the atoms. If you look at glucose and galactose, both are hexose sugars, and they're both in the aldehyde form. However, they're different from each other because of the arrangement of the hydroxyl group at the 4th carbon. It doesn't seem like much, but that slight difference can give them different shapes, which can lead to different binding activities, which can cause them to behave differently. There are 2 ways to draw carbohydrates. You were probably taught to recognize the ring structure, which is how they usually are in aqueous solutions, but know that carbs can be drawn in linear form as well, and you should be able to recognize a carbohydrate in either form. So here's an example of glucose. If you were to count the atoms, you would get C6H12O6. And I wouldn't worry about the numbering system for carbon at the moment. We're going to get to that later on in the course. So let's look at joining 2 glucose molecules together. Here we have 2 dehydration reactions taking place, one between 2 glucose molecules to make maltose, and the other between glucose and fructose to make sucrose. Notice the loss of a water molecule in order to create the bond. 2 things about this diagram. First, when 2 monosaccharides are joined together, this is called a disaccharide, and you should be able to recognize this when shown a picture. Second, the covalent bond between 2 monosaccharides has a name. It's called a glycosidic linkage. You are not required to know this in regular bio, but you do need to know the name of this bond here in AP land. When we continue to join monomers together, we get polysaccharides. These large molecules serve purposes that are both storage and structural and vary between animal and plant cells. The differences between these molecules are primarily due to the positions of the glycosidic linkages, but as far as this class is concerned, you don't need to get into those details. When there is an excess of glucose, both plants and animals have a method of storage, and when glucose is needed, it can be hydrolyzed or broken off the larger structure into the smaller monomers. Glucose storage in plants is called starch, and starch is stored mainly in organelles known as plastids. Animals also store glucose, but it's stored as a molecule called glycogen. Glycogen is mainly stored in liver and muscle cells. Both starch and glycogen represent stored energy, and this is what is used first by cells. The main structural polysaccharide in plants is cellulose, which is a major component of the cell wall in plant cells. An interesting fact about starch versus cellulose is that while we have enzymes to break down starch and therefore get energy, we do not have enzymes to break down cellulose. In fact, very few organisms have those enzymes. So when we eat cellulose, we call it insoluble fiber. It just passes through the digestive tract, scraping the sides of the intestines and stimulating the secretion of mucus to make the undigested food pass more smoothly, keeping you regular, if you will. Another important structural polysaccharide is chitin. Chitin is found in the exoskeleton, that hard outer covering of arthropods, like my cockroaches. Chitin is also important in the cell walls of fungi. And that's pretty much all I have to say about carbohydrates. The next episode in this series is going to be about lipids. So if you have any questions, please feel free to bring them up in class. So until next time, let's be good humans.